energy track. And we have two presentations for you in this session. And we're going to kick it off with the Missouri miners. Take it away.
All right. Okay. As he said, we're the Missouri miners, and our project is a synthetic biology approach to microbial fuel cell development. So we're producing electricity utilizing E.Coli. We're from Rolla, Missouri. Missouri, if you don't know, is in the middle of the U.S. And then Rolla is sort of central, maybe a little south. Our university is Missouri University of Science and Technology, or Missouri S&T. This is just a little bit of background about our university, some interesting facts. This is Sandra Magnus. She's one of 3 astronauts who have graduated from Missouri S&T. The nanodrop was co-developed by one of our alumni. He actually donated 2 of them to us. This picture here, Therosphere, is a material that a professor in our ceramic engineering department developed. It's used to treat liver cancer. This is our team's fearless leader, Marcus. Unfortunately, he couldn't be here today. And then this final picture is St. Pat's. St. Patrick's Day is a huge tradition at our university. It's one of the best times of the year there, and it's because St. Patrick is the patron saint of engineers. So, as we like to say, it's the best ever. So, a little bit about our motivation for doing this project. Our campus has a really big green energy initiative. Obviously, there's a growing green energy initiative in the world, and that's a big deal on our campus. We have E3, which is environment energy education. So that motivated us to work on this project. And then we also have a new Energy Research and Development Center. They sponsor most of the energy research on our campus, including our team. So this is their website. It tells a little bit more about why the Green Energy Initiative is important to us. So I'm going to go into a little bit more specifics for our team. We're a pretty small team. We're only about 3 years old. We only have 11 members, 11 active members. But we're getting a lot bigger. We're really trying to focus on raising awareness and creating more visibility. And 2 of the major things we're doing for that is becoming a student organization and a student design team in order to get more resources, more funding, increase our visibility. Spread more awareness about synthetic biology. Student design teams are a really big deal at S&T. We have quite a few of them, including solar car, solar house, concrete canoe, and chemical engineering car team, which we're actually working with on this project. And then we also do presentations, undergraduate research presentations every spring at the state capitol. And our university focuses a lot on undergraduate research. So a couple of our students on the iGEM team are actually sponsored by our OURE, our Undergraduate Research Opportunities Experience program. So this is a picture of our team and our 2 advisors. Nicole is going to get into a little bit more details about the project.
Okay, so the main deal of our project is basically to develop a microbial fuel cell, a biobattery, if you will. So we are taking geobacter sulfur-reducens as our model organism, and we're taking geobacter is sort of a more difficult organism to work with, being that it's anaerobic and things like that. So we're taking the electron shuttling genes out of geobacter and putting them into a more easier organism, E. coli. So our primary goal is to optimize that electron shuttle in E. coli. And E. coli already has some of the main parts of the electron transport chain, but it doesn't have the final outer membrane cytocromes that geobacter has and that is necessary for transport of electrons out of the cell and into a battery environment. So we're trying to optimize the electron shuffle, and that's our little mascot doing his electron shuffle dance, and that will light up our light bulb. Okay, so this is the process as it is found in geobacter. You have glycolysis going on in the cell, which will kick out electrons, and so the electrons will come up into the cell membrane and go to these cytocromes. And then in geobacter, it'll go out to these outer membrane cytocromes, B, E, S, and T, and they'll take those electrons and put them on the pilus, and take them up, and they will reduce the iron in the environment. For E. coli, they have a similar system. E. coli has this part right here, but they do not have the outer membrane cytocromes. Instead, they take the electrons and put them back into the membrane to a terminal oxidase. So that's basically the difference there. E. coli doesn't have these last 4 genes or the pilus. Now, our research has been inspired by the research of Dr. Derek Lovely. He's done a lot of work with geobacter, and he has proven that the pilus is actually not necessary for transport of electrons outside of the cell. So in putting our genes into E. coli, we don't actually need the pilus, which is a good thing because that's really hard. There's like 20 genes, and it's a lot. So we're not going to put the pilus in. But will this actually work in E. coli, right? That's a pretty big question. It will. Dr. Sporman has come up with this. He did some research with our OmcB gene, very similar like that, and put it in E. coli, and with that he got a very limited electron potential. And we think that with putting the other outer membrane cytochromes in there as well, the E, S, and T, that we'll get a higher potential. So it can indeed work with E. coli. We are working with the chemical engineering car team at our school, and they're going to be using our fuel cell, which you can see right here, as part of their control circuit on their car. So we have all our little parts. The bacteria will be coating on this carbon felt. And so as the bacteria are kicking out these electrons, the electrons will go onto the carbon felt, and that's going to act as an anode to the battery, and the electrons are going to go up through the copper wire and into the cathode side and produce that electrochemical gradient. The middle part here is the positive and negative ends are separated by a Nafion 117 membrane. And so that'll just allow the hydrogens to pass through. And this is just a generalized fuel cell design. Ours is very similar, except this catalyst, instead of being what it is here, it will be our actual bacteria. And so it'll just shoot up those electrons, or take them off, and the electrons go up over the wire and into the cathode side. And the protons will be separated and go through the Nafion membrane over to the other side and join with the electrons like any fuel cell and create energy, right? Okay, we haven't actually been able to test our fuel cell because we've had some difficulties. We just haven't gotten there yet. So we're looking at the research of a graduate student on our campus, Varun Paul. He has been working with bacterial fuel cells with haloalkophilic bacteria, and basically we're expecting our fuel cell to look very similar to his results. So our voltage would be something similar to this, and our current, very, very similar results is what we are expecting. So now I'm going to hand it over to Patrick, and he's going to tell you a little bit more.
Okay, so this is where the iGEM standard parts comes in. As many of you know, this is a flow chart of all the steps we would take to end up with our final result, this giant plasmid down here. So we start with a geobacter sulfur reductase chromosome, and through PCR amplification, we will extract our 4 OmcE genes, B, E, S, and T. We will then clone those into a E.Coli plasmid. So from this point on, it's a E.Coli plasmid. So we'll take those genes, the plasmid, and then we will extract the gene out after it's cloned because we need to get the DNA concentration high enough to continue on. And when we have that gene extracted, we will put it in a ribosome binding site plasmid. So this way, from this point on, this gene is now functional. So then we do this in parallel with all of the other 4 genes, and then we will take this plasmid and this plasmid, and we'll cut both. We'll cut one gene out of the other, and we'll cut the other one, and then we'll put this POMC RBS gene right after the other one. So we'll end up with a outer membrane cytochrome B, E, and the ribosome binding sites for each respectively. And then, of course, like I mentioned earlier up here, we're doing everything in parallel. So we already have this ready, and then we just repeat this step, and we end up with our final plasmid. And before we came here, we submitted our parts, and these are the 4 parts we have submitted so far. They are right up here. We submitted them right before we got done. And Erica will go through the issues we had with that later on of why we didn't get farther down. All of these parts have, of course, already been sequenced, so we all know that the correct genes are inserted. And as mentioned before, our objective is to get all 4 genes into one plasmid. And some of you are probably wondering why, and some of you probably already know that answer. It's because 4 plasmids would just not be nearly as efficient. So if we can have them all in one, it'll have a much more higher efficiency. And I don't know how many of you guys went to see the FBI guys talk yesterday with their scary demonstration about biosafety, but just to satisfy them, we are in a biosafety level 1 lab, so all of our stuff is safe, including geobacter. And this, I'm sure all of you guys already know in great detail how this works, but for the few of you who don't, this is in much greater detail, this whole thing right here. I'm going to try and break it down. So we'll have a geobacter sulfur reductase. Through PCR amplification, we will add our prefix and our suffix, and then we will go through a topo cloning step to add it to this plasmid right here, and then we end up with this plasmid with this gene inserted. So then we have all our available restriction sites for use later on for any manipulation we want to do. So we will take that gene out of that plasmid using the restriction enzymes mentioned right here into a standard iGEM biobrick that has a ribosome binding site and a promoter in it. So we will cut them, get them ready, we'll ligate them, and then now it's a functional gene. And this is one of the parts we have submitted. The other 3 are all like this. And then from there, since we have 4 of them in parallel, we will take a gene out of one, cut it the same way we did in the previous step, insert it in, ligate them together, then we'll end up with this big plasmid. And since we did it in parallel, we'll have another one just like it, but with the S and T genes. And then we will cut that out, insert it in there, ligate it together, and then we have this huge BEST plasmid. And in Rolla, for the St. Pat's festivals we like to say it's the best plasmid ever. And I'm going to let Erica wrap things up.
So, as we know with many research projects, you run into a few road along the way. One of our road bumps was due to the difficulty of working with the anaerobic nature of geobacter combined with difficulty with the Nafion membrane, we weren't able to test our fuel cells. So that's why Nicole mentioned earlier we weren't able to test fuel cells, and that's why, because we couldn't get these to work together. We also ran into a minor road bump with the PST1 site. There were actually 2 PST1 cleavage sites forming 3 fragments of our plasmid instead of the expected 2. But our biggest problem was our inefficiency with ligation and transformation. In the future, we plan to do more extensive testing and modeling of our system. We wish to determine the redox potential for each cytochrome. That way we can determine which order they will go in to have the best efficiency and improve the efficiency of our electron transfer proteins in our E.Coli. We also plan to couple Schwannella. As mentioned, as seen earlier in the presentation, it is another iron-reducing bacteria. We plan to couple that with our geobacter E.Coli in order to improve efficiency of the system. We hope that it'll make things work a little smoother and give us more voltage. So, what you've seen here today, what we've presented to you guys, is very relevant to all the projects going on currently at Missouri S&T, including our brand new doctoral research and training program in alternative energy technology. This program is sponsored by iGEM Fellowship, and if any of you happen to be interested in our program, go ahead and read some of the information on the slide and contact us after our presentation. The director of this program is Dr. K.B. Lee. We would like to give a special thanks to all the people who helped make this possible, including our wonderful advisors, Dr. Westenberg and Dr. Katie Shannon, as well as the departments and companies listed here. Those are just our references. And so we hope you guys enjoyed our presentations, and we'd be happy to answer any questions you may have. Thank you. Does this one work, Rafi?
Hello? Okay. Did you use any of the designs from the Harvard fuel cell from last year's iGEM team?
I didn't see Harvard's fuel cell from last year, so I couldn't say.
Oh, really? It looked really similar, so I thought maybe you would, because they had put up like the sort of an open source design, and I wondered if you had improved at all on the design.
I have no idea.
Okay. Well. What?
There's some.
Oh, okay.
Yeah.
Okay. And then my second question, I was wondering, you said that you had trouble with how geobacter is anaerobic and with the fuel cell. Did you try bubbling like nitrogen gas through the chamber and that it still didn't work?
Yeah, it's just it kept dying before we ended up. Wasn't that the issue when Marcus would have it shake over? Yeah.
Okay.
Just little things.
Are there any more questions?
Hey, sorry, when you get your fuel cell working, what are you planning to use as a carbon source to provide the energy?
As a carbon source?
Yeah, for the cells to go on. Are you planning to go out on wastewater or what?
What? No, it's glucose.
Yeah.
Just glucose.
Okay, so that's, well, glucose isn't really a practical substrate for producing renewable energy on a large scale. Do you have plans to sort of make it into a more practical device that could actually produce energy from renewable materials, cheap renewable materials, I mean, unlike glucose?
Would acetate work? Yeah. Acetate?
Yeah. So.
E.Coli?
Yes.
Yeah. I think geobacter does, doesn't it? I think geobacter does, right? Does this work? Yeah, it does work. Geobacter does, I think, right? Growing acetate? Yeah. I was just double checking. Yeah. I don't think so, but it might.
Well, we were more concerned with the immediate steps in our process, so we haven't gotten that far. We're, like Nicole said earlier, we're working with a chemi car, and that was our plan right now was sufficient for that process. Can you see what I'm saying? So we haven't gone further yet, but we plan to.
How big is the car? It's about the size.
The actual car is about the size of an RC car. They just took the chassis and then chassis. I keep saying that word wrong. And then our fuel cell is actually.
I had a question. So you didn't quite accomplish the one giant plasmid with all three genes. Did you get the three separate genes working in an E. coli, or any one of them working at all?
Yeah, I made a mistake earlier in the presentation when I said ribosome binding site when we submitted. There wasn't. Yeah, I made a mistake earlier when I pointed out the ribosome binding site as the parts we submitted. Can I have a clicker?
Yep.
Yeah. The part that we submitted is right here, so it's not functional. I made a mistake earlier, sorry. So it's not functional, you can't tell, because it doesn't have the ribosome binding site. Without that, nothing's really going to happen. It's just a gene just sitting there. So
that's when we were having problems with our ligation and transformation, so we weren't able to proceed further.
Any more questions?
Great, thanks a lot.
Thank you very much.
Thank you.